our speaker is Kelly Klingler. She's a postdoctoral scholar with UNR, and we're going to learn a little bit more about two iconic Western U.S. species. So I'll turn over the floor to Kelly.
Awesome. Thank you so much. Thanks for everyone for joining us tonight. Thanks to F&W for organizing and hosting such a cool speaker series. And I did too want to acknowledge that today is the 56th anniversary of the Wilderness Act. So September 3rd, 1964, I think 9 million acres were signed into protection. I think maybe nationwide we're closer to over 100 million, but I could be wrong. I'm not an expert on wilderness, but thank you to F&W for all the work you do to preserve these wild places, for the wildlife species that I'm going to talk about today. So I'm excited to share some of this work that I've been engaged with as a grad student and now as a postdoc and lab manager at UNR. And I chose this photo to the right because I think it really sets the stage for the landscapes that we're going to be talking about today. This was taken in the Sweetwaters just a couple of weeks ago. And these are the rugged areas, this alpine topography that really host the animals that we're going to talk about and that has contributed to the population level resources we have in terms of their conservation efforts. So as a conservation biologist, I think a lot about variation and the variation that we observe in nature, specifically the variation that occurs within a species. So while you're out recreating or hiking in nature, you often hopefully see wildlife while you're out there. Sometimes individuals within a species look pretty similar, and sometimes they can look really different from one another, like these Hawaiian land snails on the upper left. That's true for both wild populations as well as domesticated species, right? There's a lot of physical or phenotype variation in our companion animals. And so I've always been really curious about the genetic component, this hidden underlying population level resource that really is so important for conservation, because that's the scale at which many of these biological processes operate. So individuals don't go extinct, but we can have populations collapse. And if you have enough populations collapse, eventually you can have species level extinction. So I've always been really curious about characterizing genetic resources at this scale. And as I mentioned, it's really because the way that we generate new variation in a population happens across four basic mechanisms. And this cartoon really depicts that. In the upper left, we have mutation, and this is really one of the few ways you can introduce novel or new variation into a population. It's the physical altering of the DNA sequence data. So at some point during replication, the DNA makes a mistake, and we have an A base substituted for a T, right? Something like that. And sometimes this has downstream effects and we see a physical trait difference. Here it's led to a brown invertebrate, but it doesn't always lead to that sort of physical trait that we can observe in nature. To the right, we have another mechanism. This is gene flow or connectivity. And so you can imagine that if one individual migrates to a new population away from its natal site, and it successfully finds that population, establishes and reproduces, it can then pass its genes to the next generation. And so all of a sudden we have a population that was once all green invertebrates that now has some representation from the brown morphotype. A third mechanism here in the lower left is called genetic drift, and that's for these purposes kind of jargon, but it's just meant as a chance event. So you can imagine that here, just by bad luck and random chance, all of the green invertebrates suffer a mortality event. They all get smushed, not because they were less adapted to conditions, but just because they happen to be in the wrong place at the wrong time. In nature, this often occurs through things like severe weather events or unpredictable events that can take out a portion of the population and therefore a portion of the gene pool. Finally, we can imagine that if you have a predator like this chameleon who, let's imagine, can see the green invertebrates a little more easily, we know that the green invertebrates will then be predated upon more frequently. And so it ends up being advantageous to be this brown morphotype. And again, the population shifts, the frequency of morphotypes shifts to be mostly brown because it's advantageous in this environment, in this population, to hold that characteristic. At the end of the day, I think the main take home here and why we care about population genetic variation is that it enables species to adapt to changing conditions. I kind of liken it to this little boat in the lower right, that if your boat is well stocked with food stores and water and perhaps life vests and lifeboats, then you're better able to weather the storm. You're better able to weather these unpredictable events. When you have fewer population resources to pull from, you may not have the right combination to adapt to a certain new environment. And unfortunately, we live in a time when a lot of our population level resources are at risk, primarily because of anthropogenic disturbance. So habitat fragmentation across the landscape, introduction of invasive species, noise and light pollution, and of course, climate change. So there's evidence that the Earth's climate responds to changes in greenhouse gas levels that can be found in tree rings, ocean sediments, coral reefs, and it appears that current warming is happening at roughly 10 times faster than the average rate of ice age recovery warming. In this figure on the left, you have annual and decadal averages of temperature increases up through 2010 or 2012 in this case. And there are many metrics of climate change. This is the decline in sea ice, and you can see an increase then in sea level change. And because a lot of us spend a lot of time in these alpine environments, in these mountainous regions, you know, they appear really pristine. They're areas that still hold wild places that are not affected by residential development or agricultural runoff, for example. But despite appearing pristine, some of the evidence suggests that climate change could really affect mountain ecosystems. So similar to how regionally climate change will differ, the Pacific Northwest, for example, in the US will have very different effects from climate change than the Northeast. Similarly, rainforests are going to respond really differently than alpine ecosystems. There's some evidence from the Northern Rockies that higher elevation sites are warming at 3 times the rate of lower elevation sites. But given that, we should also acknowledge that mountain environments are really rugged, they're really topographically complex, and because of that, some organisms, those that are able to disperse, may be able to move relatively short geographic distances, perhaps into the next drainage, right? And all of a sudden have a north-facing aspect or some cold air pooling that can really make conditions much more favorable for that population to persist on the landscape. So we'll think about that in the backdrop of 2 really amazing species from the Western US, the American Pika here on the left, which is a small mammal species that inhabits these alpine talus slopes. And then on the right, we have the Lahontan Cutthroat Trout, a game species and a fish species that occurs in these riverine and lacustrine systems. So we'll start with the American Pika. Here on the left is their broad distribution map across the Western US. You can see 5 pretty large oblong circles there, and that's representing 5 distinct lineages within the American Pika, 5 subspecies. And you can see that they're kind of each associated with a specific mountain system. So we have the Northern Rockies, the Southern Rockies, the Cascades, the area we're going to be talking about today, the Sierra Nevada and Great Basin, and the central Utah. And I think this is why pikas are really popular, right? Wherever you are in the Western US, you probably have the chance of getting to see these guys while you're hiking. They are herbivores, so they eat vegetation, and they're considered to be lagomorphs. So lagomorph is, it means they're basically highly related to hares and rabbits. Because they're a rabbit, they don't hibernate. And so they are aware all year long, and that requires 2 diets. So they really forage for an immediate diet, their immediate needs, and then all summer, they're caching vegetation for this delayed consumption, this overwintering food source. Here in the lower right, you can see a hay pile. I took this just a couple of weeks ago in northeastern California. This guy's been very busy and very proactive in getting his cache ready for the winter. They're not always this showy. They're not always on the surface and visible. Sometimes, and more often actually, pikas will stuff that into the interstitial space or the in-between space within their rocky substrate. And this pika isn't putting this hay pile just anywhere in this rock. Each individual, male and female, defend territories within the rocky substrate, whether that's a boulder field and high elevation habitat, or perhaps lava flows and lassen, or anthropogenic ore dumps. It doesn't always matter exactly what the substrate is, as long as it buffers the ambient conditions. So because pikas enjoyed much broader distributions during the Ice Age, during the Pleistocene, when we had a lot of glacial and interglacial periods, they're really adapted for cold conditions. In fact, their body temperatures are within 3 degrees of their lethal limit. So these guys are very, very prone to heat stress, and so rely really heavily on this substrate just to thermoregulate. In addition to being territorial, they're also vocal, which is kind of weird for a rabbit. They produce short and long calls, and they're also asocial, so they really only tolerate one another during the breeding season. Here are some more fun pictures of pikas. We have a guy here on our left moving through some rock. He's calling out at you. This is a pika from the Ruby Mountains, right before we got driven out by the Harrison Pass fire last summer. You can kind of see it's hazy and orange in the video. But these guys are really fun to work with because you often get to spot them. They're not an elusive animal. Because they're territorial, they're often out calling at you when they occur in places. And here on the bottom are some other behaviors, one caching some vegetation, one calling, and one behaviorally thermoregulating. So I mentioned that we were going to zoom in on the region in our own backyard in the Great Basin. And this is a particularly important region for pikas across their distribution because it's really the region that has experienced the highest rates of pika loss. So here in blue outlines the hydrographic Great Basin. It's cut off here at the bottom for some modeling reasons in the paper. But the general idea is that this is the region where across time we continue to see declines. So the Great Basin pikas are very fragmented in nature, just naturally. The mountain systems within the Great Basin are kind of sky islands. Mountain ranges are very separate from one another, and so the small mammal communities they host are also very separate geographically from one another. You can see here on the right, we have sampling region on the X and percentage of historical sites extirpated or collapsed on the Y. And you notice they're colored differently. So the black bars here are representing pika population collapses just between 2014 and 2015. The gray bars are representing pika populations going extinct in the 20th, or since 1994. What's important to remember with the Great Basin is because this is a region that occurs in the southwestern lower latitude of the range, and because of the nature of this fragmented mountain system, pikas here have been retreating upslope probably since the start of the Holocene, probably since 8,000 years ago, 10,000 years ago, as we moved into our current interglacial, the Holocene, we've lost pikas. The concern is that within the 20th and 21st century, we're seeing rates of extinction like never before. Here in 2014 and 15, we see a complete collapse of the Zion population. If you're curious about pikas in Nevada specifically, I've put up this slide. So on the left, we have a figure just showing sort of the outline of the state of Nevada. The open circles here representing occupied sites, the dark circles representing sites that were historically occupied by pikas, so like 10,000 years ago, and then sites with crosses that were occupied up until the 20th century. Here on the right, I've listed ranges in case you were curious about a certain range and whether you could maybe find pikas there. And I've categorized them into 4 different categories. So the first are mountain ranges with current pika sites. So they still support pika populations. That's not to say that They haven't experienced some population collapses, but as a whole, the range still supports pikas in some locations. The second category are ranges where we've documented sites collapsing, populations collapsing. This is not an extensive list, but it is specific to Nevada. And I've highlighted ones or placed asterisks in ones that were reported to have pikas up till like the 1930s. The third category are not historically reported and not currently detected. So these are ranges where no one ever submitted a natural history collection in 1920 or 1910, and we don't see them there now. The fourth category is sites where we have actual skeletal or fossil remains indicating that, yes, pikas, conditions were suitable or favorable enough to support pikas that far back. So let me know if you have any questions about certain ranges. I'm not an expert on Nevada pikas in general, but hopefully that's a little bit helpful. Here on the lower left, we have a quote from Hall, who in 1946 produced the first survey of mammals in the Great Basin. And he indicated that, you know, pikas are probably really much more widely distributed in northwestern Nevada than what we have. We only have 3 records. And so northwestern Nevada was generally unexplored in terms of pikas, or at least underdocumented, until 2017, when a group of researchers explicitly went out into this area and surveyed over 200 sites to get a sense and finally put in the scientific record whether pikas had existed here. In general, they found 2 times, they found sites, they found most of the ranges had occupancy, but there was a lot of prehistoric or relic sites that had former pika occupancy but don't currently host pikas now. So they found 2 times the number of relic sites than they did currently occupied, which suggests that this region is also experiencing quite a bit of a decline in population persistence. For example, here in the lower right, we have the Black Rock Range, and you can see that these are all dark triangles, which represents collapsed or extirpated populations. So despite the Black Rock really representing the highest elevation and coolest temperature-wise range, it wasn't able to continue supporting pikas. This is in contrast to the Home Camp Range, one of the lower elevation warmer sites that has some stars here, and these stars represent currently occupied sites. So I think it's important to drive home that there are a lot of complexities and nuances when you're dealing with a small mammal like the pika that experiences its microclimate and microhabitat at really small scales. So it may be that the Home Camp Range is at lower elevation, but potentially it has better airflow through it, the substrate buffers the thermal habitat better. There's something about that site that enables persistence. And so we have to be careful about making generalizations with pikas because of that. They continue to persist in locations that we're surprised by. Before we go into what we know about their population genetic resources, I just wanted to provide this little cartoon as for some context. As I mentioned, the Great Basin region is just fragmented by nature. Most of the mountain ranges are really separate and distinct, and they form kind of islands unto themselves, at least for the small mammal communities that occur there. Unlike birds that can kind of disperse between mountain ranges, that's not going to happen for a pika. It's not possible for them to move down the elevational gradient, cross the desert valley, and then move up again to another mountain range. So we can imagine here that if you have a pika population on a peak that has a good number of individuals, and it has some connectivity with a population maybe a kilometer away, then we have that gene flow happening. We have that connectivity happening. And so population variation is being maintained. And so you have more in your boat, more resources to persist into the future. This is really in contrast to this population that has many fewer individuals and does not experience any contact with other populations. And so may randomly lose variation, as we talked about, or could have some problems associated with inbreeding and relatedness. So I think this helps kind of just think about it in terms of populations in the Great Basin. So to get into some results and some data about pikas, I was part of a group that studied 11 sites across the Western US, in Montana, Colorado, Nevada, and California. 3 of those 5 oblong shapes, those 5 subspecies were represented here. So we have the pink areas representing the California subspecies. We have the Ruby Mountains and East Humboldt pikas that we're going to focus in on. Montana populations, so these 2 are actually the same subspecies, if you can believe it. They experienced some connectivity probably during the last glacial maximum. And then the subspecies within Colorado. So to get kind of really high resolution genetic data, to get genetic data that represents the full genome, maybe not the full genome, but a good portion of the genome, we have to collect high quality tissue. And so individuals were live trapped. We've got a guy here in a tomahawk live trap, and we just take a little bit of ear tissue and let them go on their way. These guys are pretty difficult to trap, so if you have any questions about how to handle how pikas are to trap, I always have stories about that. Some of the results here that we'll zoom in on, just if we look on the left-hand side of this table first, the main point I just want to drive home, we have population on the left, the number of individuals sampled, and then a measure of genetic diversity here on the right. It's nucleotide diversity. In general, you'll notice that all the pika populations kind of have limited amounts of genetic diversity. And that's true for mammals in general. Compared to other taxa, they seem to have lower levels of genome-wide diversity. But if you focus in on this purple circle, the 6 Nevada populations, you'll see that they have an order of magnitude fewer genetic resources compared to their California, Colorado, and Montana counterparts. And that's really concerning. It suggests that their boat does not have enough resources potentially to adapt to changing or differing conditions. On the right here, we have a figure where each individual is plotted, and they're plotted in terms of their genetic differentiation and their genetic similarity to one another. So you can see that all the California pikas are kind of clustering together over here. All the Colorado individuals are clustering together over here. And the further apart these clusters are from one another, the further differentiated they are, the more genomically distinct they are from one another. And you can see here that, of course, the Montana in brown and the Nevada populations in purple are a little closer because they are the same subspecies. But even being the same subspecies, they're very strongly differentiated from one another. You'll also notice the Nevada populations has less spread, right? So despite sampling 6 populations, all of the individuals look pretty much the same. Those circles are almost plotting one on top of each other, compared to, for example, in Montana, where you have some differences between populations. I think the main take home with some of this is that you have the problem of small populations, and it's likely not just pikas, but there's probably other small mammals where you have really long-term geographic isolation, a really limited dispersal ability. Your life history just isn't equipped to go more than 2 kilometers, 3 kilometers in a lifetime. And so over time, you start to lose some of your genetic diversity. And it's possible that your population will persist anyway, that the substrate might be enough to get you through, but it's also possible that we may randomly lose that population in an event, or perhaps they're not able to adapt to changing conditions. And I think that's really striking when you think about an organism that actually occurs in wilderness areas, occurs in wild places where it's still pristine. This isn't an area where we're dealing with a 7-Eleven on the corner, but it does mean that you can lose organisms and you can lose populations even within pristine habitat. If it doesn't function the same way as it used to, if it doesn't buffer the climate in the same way it used to, you're going to still lose populations. And unfortunately, for organisms like pikas, we don't have a lot of management options. So pikas are not in zoos. They're not able to be bred captively. They die of cardiac arrest before you leave the talus slope. So it's not as if we have the options to sort of bring these guys in. And it's hard to watch some of these populations continue to decline in areas of the Great Basin because of that. We have a new threat as of this spring. The rabbit hemorrhagic disease virus 2 has entered the US from Europe, and it was confirmed in wild rabbit populations in April. First in the Southwest in New Mexico and Arizona, but I'm aware that this has spread to California as well as I think Texas. And it doesn't discriminate among rabbit species. So a black-tailed jackrabbit as well as cottontails have been found dead. It's highly lethal, and there's no real reason to suspect that it might not affect pikas. And so we have this whole novel disease layer that we are also dealing with in terms of the conservation of this organism. That being said, there's also always good news, right? On the left, we have the illy pika. This is colloquially known as the teddy bear pika, and it was discovered in 2015 after more than 20 years of never being seen. And this pika occurs in northwestern China. Most of our pika species globally, the diversity of pikas occurs in the Tibetan Plateau in the northwestern China. There's about 28 species there. And so that was really exciting. And it really just relates to the fact that we don't know all of the complexities going on. And as I mentioned, there might be some populations that continue to be resilient. You have site-specific idiosyncrasies that may help provide some climate refugia. I think if you're a population that has some connectivity to the mainland stems of the Northern Rockies or the Sierra, you'll probably fare better. And then being a pika, they have short generation times. They're a rabbit, so they might be able to recover more quickly than we give them credit for. So with that, I will shift gears and talk a little bit about the Lahontan cutthroat trout now. A very different species, but one that has been tremendously affected by the physical topography in the Western US as well, but which has a different conservation story because of its ability to be raised in captivity. So here's a little GIF that I have showing the ebbs and flows of ancient Lake Lahontan. So ancient Lake Lahontan covered most of northwestern Nevada during the Pleistocene. It reached its high stand around 600,000 years before present. And it desiccated over time, kind of to near present day levels around 8,000, 9,000 years ago. And you can imagine that Lahontan cutthroat trout populations would have experienced really varying levels of connectivity over that time. And so you would have similar kinds of periods of isolation like pikas have on their mountain sky islands. So when we think about the Lahontan cutthroat trout and the population genetic resources that it has, we really have to start over here in this figure. So on the right, we have some of the subspecies of cutthroat trout that evolved with ancient Lake Lahontan and in the Western US. And I think as of today, there are 14 recognized cutthroat trout subspecies. And here are some of them indicated here. We have coastal, Yellowstone, and the ones we're talking about today are the Lahontan cutthroat trouts, what's in this neon green area. But it's not to say that these other cutthroat trout species don't have their own conservation stories and issues. It's really tremendously variable what these cutthroat trout look like, and they've all evolved differing food, prey items, differing morphology, differing sizes. It's pretty incredible that we have such diversity, but the Lahontan cutthroat trout was the largest of them all, and is currently the state fish of Nevada. It existed in this Lahontan Basin since at least the end of the last ice age, again, that 10,000 year mark before we entered the current interglacial. And it was found in these cold water habitats, whether they were lakes or rivers or streams. There's a lacustrine or lake form of the Lahontan cutthroat trout. And it really supported a tremendous inland trout fishery until the 1930s, up until mid-century, 20th century. And unfortunately now, LCT have been extirpated or are locally extinct from more than 90% of their historical habitat. If we zoom in on that neon area, this green Lahontan Basin area, there's even further population structure. So biologists recognize 3 distinct population units within the Lahontan Basin. We have the Eastern Basin Humboldt Lahontan Cutthroat Trout, the Quinn River Cutthroat Trout, Lahontan Cutthroat Trout, and the Western Basin. And we're really going to focus on the Western because it's what's in our backyard, but it's also the most imperiled. It has the fewest extant or current occupied sites for LCT. I've listed here what we have left within the Truckee River Basin, within the Carson River, and the Walker River. The fish that you fish out of Walker River now and in a lot of the Truckee River Basin are maintained by hatchery propagation. So the Walker River largely collapsed by the mid-20th century, and our recreational angling opportunities and fisheries are managed almost all by hatchery propagation. I think to put it in context, in 1844, there were 11 lakes and between 400 and 600 streams that supported LCT. Nowadays, we have just 5 lakes and 120 streams, and only 10% of those streams are self-sustaining, meaning it will always require a hatchery stock to be seen on the landscape. Historically, LCT, it's a phenomenal story. It migrated 120 miles from Pyramid Lake to Lake Hot Tahoe via the Truckee River. That was their spawning run. Anytime between February and July, male and females would travel that, and that was how they reproduced. These fish were so big, they were considered and called the monster salmon trout. We have historic records of over 40 pounds for fish. But as I've mentioned, by the mid-20th century, by the 1940s, fish had largely vanished. And that's due to a lot of reasons. One being water diversions for agriculture and ranching and other things, barriers and dams. I think currently on the Truckee River, there are at least 41 in-stream structures to this day, including Derby Dam. So fish are no longer able to use the habitat in the way that they evolved to use that habitat. And so they cannot be self-sustaining without being able to have that connectivity restored. We also had issues with non-native and still have issues with non-native fish. So we have competition for food resources and predation by non-native brook and lake trout in most of our waters. We have the issue of hybridization with rainbow trout, and that's an issue and concern because you have 2 distinct gene pools that then become mixed and potentially swamping out some of the distinct variation of both of those species. And so you end up losing 2 species and gaining 1. And then of course, all the commercial overharvest that took place for LCT and for a lot of species in the 20th century. What I'll note here is that the reason we have a lot of these introductions of invasive trout and species was not out of ill will. A lot of 20th century folks felt they were doing a service by moving fish around. So you had bucket brigades where people would just, wherever their favorite, fishing holes were, they would then take those fish and move them upstream or move them to a different location. And they felt that they were enabling some recreational fishing, but also allowing people to support themselves on a local food source, and not realizing the disruption to the ecosystem that would have. And so this is truly a story of how population genetic tools helped us to piece together how to recover this historic and threatened fish, which was designated by the Endangered Species Act as threatened as of 1975. And it's really in the 1970s that we see a shift in thinking about species on the landscape, and for the LCT, it was transformative. This man here, Robert Benke, was a prolific fisheries biologist during that time, and he happened upon a stream in the Pilot Peak Mountains, so right along the border of Nevada and Utah. And when he saw the fish that occurred in Pilot Peak, he felt that they looked morphologically similar to what he knew the original LCT in the Lahontan Basin looked like. At that time, they didn't quite have the tools developed yet to really be able to test that. But by the 1990s and early 2000s, we've been able to go back to museum mounts. And this is work that predates me, but that I can share a little bit. The population genetics were done of museum mounts. So they went back into the museum and natural history records all the way back to 1870, where fish were caught in known locations. And then they compared that to the genetics of the pilot peak fish and found them to indeed be descendants, or as close to the descendants of the original Truckee Basin LCT as we could get. And so I think there are two main take-homes here. One, this was a result most likely of somebody moving fish at some time during the mid-20th century. It's possible it was one guy that moved 3 fish. Maybe he did it 2 or 3 times. Maybe the fish were siblings, right? Regardless, it doesn't likely represent the full genetic pool of the ancient LCT. It represents a small portion of what once existed and what was adapted and had evolved within this system. But regardless, it was a tremendous opportunity to be able to do something to bring this species back on the landscape in a way that is not possible for a lot of species. And so hatchery efforts were started at a lot of levels, at tribal, state, and federal levels. I work primarily with the Lahontan National Fish Hatchery in Gardnerville, and they manage a brood stock or a stock of these pilot peak LCTs, this genetic strain as close as we can get to what once existed. And it's a lake form, it's a lacustrine form. So they raise a ton of fish every year. Each fish is individually pit tagged so that it can be tracked spatially. And then genetically analyzed in the lab, and that's kind of where I come in. And then individuals are paired in a way that preserves and maintains the diversity that we still have. This is a little bit of what that looks like. So we receive hundreds, usually 600 to 1,000 fin clips in the fall. And then we isolate that genomic DNA in the lab. We extract DNA from those fin clips. And then we genotype all the individuals at diagnostic and diversity-based loci. So we get a sense of whether we need to be concerned about any hybridization with rainbows or other invasive trouts. And then we also just get a measure of the population level diversity and the individual diversity. Once we have those genotypes, we're then able to estimate some genetic parameters. So we're able to get a sense or an estimate of genetic similarity among individuals. We can account for rare alleles if an individual happens to harbor an allele that we only see at a frequency of 10%. And then we end up generating this mating matrix that can inform for the broodstock managers who to mate with who. And because you have a limit on the amount of resources within the hatchery, a subset that maintain that pool are left as production stock, and the rest are then released for angling opportunities. Over time, if you look over here on the right, we've seen increases in genetic diversity. Allelic richness is a measure of genetic diversity over time, at least since 2008. And we've also seen increases in the effective population size, or it's kind of the breeding population, the individuals that are of breeding age. And so those are really good signs. We also continue to receive reports from anglers that fishing in places like Pyramid Lake here have really improved. So this is a photograph of Brandon Reeder, who was an undergrad in the lab with me, working on the genetic analyses, but he has also worked extensively for state and federal agencies collecting these samples for us in the field. And he caught this guy in January of this year in Pyramid Lake. And so we're starting to get to these places where we're getting to the monster salmon trout again. These are 30 to 40 pound fish. This is the pilot peak strain. It was released into Pyramid Lake, I believe, in 2006. And it takes time for these fish to mature. But we are finally starting to see some of those effects, and anglers will share that, even a few years ago, a 10-pound fish was a great catch, but now we're getting ones that put up a way bigger fight.
Kelly,
Yeah.
going back to your previous slide, you had a question from Facebook. It was, how long does it take you to genotype the fin clippings?
Oh my gosh, it's a great question. It takes a long, long time, honestly. So the process takes probably in full, if I get the fin clips in August or September, they're not done until December. So just the sheer number of samples means that it takes quite a bit to extract. They're extracted in 96 well trays at a time. And we can do maybe 3 or 4 trays in one extraction. And then your PCR, this system for getting the genotype itself, can sometimes fail and you have to do reruns. And so it can take a long time, but for me, it takes, 2 or 3 months. But by December, I have the mating matrix and that's sent to the broodstock managers. Cool. Okay, almost done here. So I am just going to play this little video on the right and hopefully you guys can see that. Sorry if it's loud, I'll turn it down a little bit. In 2014, the team really responsible for getting these guys back on the landscape and back in our rivers and lakes observed pilot peak cutthroat trout spawning in the lower Truckee. And this is the first time that's occurred in over 80 years since they largely vanished. So this is incredible. It's not their full obvious historic migratory route, that full 120 miles, but it does suggest that they're starting to see the behaviors that once occurred. They're starting to see an opportunity or a glimmer of hope that we might actually be able to have self-sustaining populations. You can imagine that to rely on hatchery propagation year after year and it is not sustainable in and of itself. It costs tons of money, and you can have like a collapse of the production stock in the fish hatchery if a bad bacteria gets out or if the water flows aren't right. So it's a tricky system. And so this was the first time in 2014 that it looked like, okay, we might be able to get these guys back in a way that doesn't require us dumping fish every year. I think we're a long way off from that. And I think it's going to take an incredible amount of habitat restoration along the Truckee. It may never happen in full, but we do have some great markers within the last few years that indicate some real achievements for LCT conservation. Here in the upper right, we have another 40-pound guy who's got a tui chub in his mouth. So these are very exciting to see. I'll end here by just emphasizing the power of partnerships. So I'm a very small part and a very recent addition to a team that has been working on this since the early 2000s and late 1990s. And it's always been so impressive the way that you have nonprofit organizations, state and federal agencies, tribal leaders, and academic researchers collaborating together in the name of species and organismal conservation. And this is one of my favorite quotes: if you want to go fast, go alone, but if you want to go far, walk together. And I think in conservation stories, the network of people that benefit, I mean, it's not just the fish that benefits from getting back into historic waters, but you have undergrads that get some training in the lab, that get to volunteer and take independent study credits. I get the opportunity to learn new genetic assays. And so there's this whole network of opportunity and benefits that come with conserving these wild species on our landscape. So with that, I just want to acknowledge all of the people that are always involved behind these conservation efforts. And there's my Pika hike team in the lower left. And with that, I thank you all for coming tonight and bearing with me, and I'll take any questions.
Yeah, feel free to unmute yourself or type into the chat on Zoom or either Facebook. And I'll get us started with a couple of comments from Facebook.
Cool.
So Gregory Kinney loves the presentation. So you have 5 stars from him.
Oh, thank you. That means the world. Thank you.
And he also recalls seeing Pika while he was backpacking. He gave me an answer. in the Sierras.
Oh yeah.
He also asks, are there any near Whitney?
I believe so, yes. And my husband actually hiked that one year. And yes, I think there are pikas there. I haven't personally seen them, but the Sierra Nevada in general, too, have a ton of pika populations. Northeastern California is a region that has also experienced some loss. I've seen between 15 to 20% estimated of rates of loss in that area. So less than the Great Basin, but we certainly lost them there too.
And Red Rock Naturalist on Facebook asks, what do you see for this species in the next decade?
For the pika specifically, or?
Yeah, but let's apply it to both though.
Okay. For, the pika, they're an interesting contrast just because we have so many more options with the LCT in terms of being able to propagate these guys and actually move them in trucks and feed them through tubes into lake and river streams, right? So it's a whole different ball game. Whereas pikas are very difficult to live trap. Once you have an individual in a trap, you then have to put them under with anesthesia. They're not like other small mammals in that regard. They're not hardy to handling. And so to imagine catching one and then trying to hike it down and get it in a car into a facility is pretty overwhelming to think about, and I think would have some high rates of mortality. There was one individual that did it in Durango in the early 70s, and there was one individual in Berkeley that I'm aware of that did it in the 50s, but with lots and lots of mortality. And it never self-sustained itself. Those pikas all eventually succumbed. So for the next 10 years for pikas, I think we'll continue to see declines in the Great Basin and the Sierra Nevada. I think there will be strongholds in the Northern Rockies, in the Cascades. And I think there has been some talk of relocation, but it's a limited endeavor, I think, at this point. And then in terms of the LCT, I think it's really exciting. I think we're developing some better high-resolution genome-wide SNP assays that are going to give us an even greater understanding of the variation that's left in this species. And I think with all the reports coming from Pyramid, this pilot peak strain, you can see how when you put back the gene pool that evolved in that system, they do so well, right? Those are huge fish. Prior to the pilot peak strain being in Pyramid, we had what is called the contemporary Pyramid strain, which was a mix of fish from, I believe, Carson, Independence Lake, and Summit Lake. And so they never did as well. They never reached these sizes. That gene pool didn't evolve in Pyramid Lake. And so you can really see how important population level information is. I'll also point out that I know Summit Lake, which is north of the Black Rock, has a self-sustaining population of LCT. It's fed by Mahogany Creek, and that's an amazing example of one population that's still hanging on.
And Lou, I see your hand is raised. Did you want to ask your question live? I also see you typed in the chat.
Can you hear me?
Yes, sir.
Yeah.
Ah, great. Well, hey, I can't believe you let Jake go to California, but
I know it's hard. It's Nevada only.
I know. No, just a question. How do you develop parallel tracks of research on pikas and on Lahontan Cutthroat Trout that don't seem to have a ton of overlap other than some geography, but maybe not. I don't know. You tell me.
Yeah, it's a great question. So as a postdoc, I work on a lot of projects that don't actually have a lot of overlap. I also work on a system in the Andes. It's neotropical avian host, and they're lice. They're obligate permanent lice on them. And so we think a lot about population genetic variation with two species that are thought to have co-evolved together, the bird and their lice. So it's more the overlap is in the population genetic tools, in the molecular assays, in the bioinformatics that we do to analyze these data rather than the species. So as a conservation biologist, I'm always interested in species that are at risk of loss or have already experienced great rates of loss. And so I've gotten lucky to be able to work with these two species, but it's, you know, the overlap isn't obvious other than we use similar tools genetically to help their conservation.
All right. Any other questions? We're nearing an hour, so definitely don't want to hold you guys too much longer. And definite thanks to our speakers. So, any last minute questions before we wrap it up and let you guys get to enjoying your Labor Day weekends?
Yeah.
All right, and I'm gonna go ahead and scroll through Facebook one more time.
Cool.
Sure we're not leaving anyone out over there. So other than that, I see a few thank yous out there. You have a thank you from Hermie, Megan, the Red Rock Naturalist, says thank you to you and your team, and also adds Pika Power. So
Yeah, nice.
so on that note, thank you, Kelly, for speaking to us. Thank you everyone who came out on Zoom and also on Facebook.
Yeah, thank you.
Definitely. You guys have a great weekend. See you, Kelly. Thank you.
Bye, Shylin. Thank you so much.
You're
Thanks,
welcome.
everyone. Have a good weekend.
Bye, guys.